Good morning, students. This is your Hardcore 2 class. In the last class, we have studied about cell division in that mitosis, and in this class, let us study meiosis first. So first, let us know what is meiosis. So it is defined as a type of a cell division where the cell undergoes two divisions. So in the meiosis, cell is going to undergo two division, and it involves two cytoplasmic divisions and one chromosomal division, and finally results in a 4 haploid cells. So in the meiosis, we are going to observe the result as a 4 haploid cells. And the meiosis is commonly takes place in a germ cells, and hence it is called as a germinal division. And during a meiosis, the chromosome number is reduced to half of the total, hence it is also called as a reduction division. So due to the reduced number of a chromosome, that is half of a chromosome number is going to reduce, so it is also called as a reduction division. And the cell formed by a meiosis are different from that of a parental cells, both morphologically and also genetically. So hence the meiosis is also called as a heterotypic division. So here the individuals are differ from the parents morphologically and genetically. And meiosis is a type of indirect cell division involving many sequential stages, and the term meiosis was coined by J.B. Farmer, and the term meiosis is derived from a Greek word meom, which means to reduce. And based on the place where the meiosis is going to occur, it is also classified into 3 types. The first one is the sporogenetic. It means the meiosis takes place during the formation of the spore. So if the meiosis is going to take place during the formation of the spores, then it is called as a sporogenetic. And second one is the gametogenic, or it is also called as a gametic meiosis. So it is a type of a meiosis which takes place during the formation of a gametes. And the third one is a zygotic meiosis. So it is a type of a meiosis takes place during the germination of a zygote. And the cell which is going to undergo meiosis is called as a meiocyte. So meiocyte is a cell which is going to undergo the meiosis. And the meiocytes are of a different types. So first one is a oocyte. So these are meiocytes which are going to produce ovum or egg. So if the meiocytes are going to produce ovum or a egg, we call it as a oocyte. So second one is the spermatocyte, which is going to produce the sperms. And third one is a sporocytes, and which is going to produce the spores. And here we are going to observe 2 types of spores. First one is the microsporocytes, and the second one is the megasporocytes. So microsporocytes, they are going to produce the microspores, whereas the megasporocytes, they are going to produce the megaspores. So in a meiosis, 2 gametes called a male and also female, which are fused to form a diploid zygote. So the meiosis after the fertilization once again results in a haploid cells. And in a man, the diploid number of chromosomes, that is 46. Diploid number of chromosomes in case of a man. When the meiosis takes place, 4 haploid cells are formed. So here you can observe 4 haploid cells are going to form. All the 4, they are going to carry 23 chromosomes. And the process of meiosis can be classified into two division, that is meiosis first and meiosis second. The meiosis first is actually heterotypic division. So the meiosis first is a heterotypic division where two cells are formed and are of different from the parent cells. The meiosis first is further divided into four stages called as a prophase first, metaphase first, anaphase first, and also telophase first. Like that, the meiosis second is also divided into four stages, that is prophase second, metaphase second, anaphase second, and also telophase second. So first let us discuss about meiosis first. So it is a heterotypic division. In this stage, the actual reduction in the number of chromosomes are going to take place. So the meiosis first comprises of four stages, that is prophase first, metaphase first, anaphase first, and also telophase first. So before entering into the divisional stages, there is a presence of a resting stage and it is called as a interphase. And the important events of a interphase are, here there will be the synthesis of a DNA is going to take place, and also there will be the synthesis of a RNA, and also many enzymatic proteins are formed, chromatin in the form of a network like structures we are going to observe in the interphase stage, and also nucleus is under resting period without the division. So in the interphase, the nucleus is always in the resting stage. There is no division is going to take place in the interphase. So you are going to observe in the interphase stage presence of a nucleolus and a nuclear membrane and a thin chromatin materials. So let us discuss about the prophase first. So it is a longer stage in the meiosis, hence the prophase first is classified into 5 substages. So prophase first, it is a very longer stage in case of a meiosis, and it is having a 5 substages. So first one is a leptotene or leptonyma, second one is zygotene or zygonyma, third one is a pachytene, fourth one is a diplotene, and a fifth one is a diakinesis. So these are the 5 substages in prophase first. So first one is the leptotene or leptonyma. So important events of leptonyma are as follows. So first one is the nucleus enlarges in size. So the nucleus is going to enlarge in a size, and a chromatin is transferred into chromosomes, and the chromomere very long, thin, slender, and appears to be in the form of a single threads. And the longer slender chromosome move towards the nucleus and gets contact with the nuclear membrane. So this is the nuclear membrane, and the slender chromosomes, these move towards the nucleus, and these chromosomes, they are going to contact with the nuclear membrane. So in some of the cases, all the chromosomes move towards a nuclear membrane by leaving a gap at the center, and this stage is called as a synesis stage. So when the all chromosome move towards the nuclear membrane, they are going to leave a gap in the center, and it is called as a synesis stage. In the beginning, the chromosomes appear as a woolen ball, and this structure of a chromosome is called as a spireme. So the woolen ball like structures are going to form, and it is called as a spireme. So Next, second substage is a zygote or zygonyma. So the zygote play an important role in the establishment of future genetic recombination, and the chromosome at this stage are a long, thin, thread-like. So in the zygote we are going to observe the thread-like chromosomes, and homologous chromosomes from parental side and a maternal side come together and arranged in a pairs. So you are going to observe in the diagram both paternal and maternal chromosome, they are going to arrange in a pairs, and the process of pairing of a homologous paternal and a maternal chromosome is called as a synapsis. So pairing of a chromosome is called as synapsis. So H.D. Darlington in the year of 1985 is a British cytologist, and he is going to recognize three types of a pairs. So the pairing by H.D. Darlington is of three types. So when the pairing starts at the terminal region and move towards the centromere, it is called as a pro-terminal synapsis. So if the pairing is going to occur at the terminal or end region and it starts towards the centromere, it is called as a pro-terminal. And when the pairing begins at the centromere, so when the pairing begins at the centromere region and move towards the terminal region, so it is called as a procentric synapsis. And the third one is random type. So here the synapsis starts at any part of a chromosome other than a centromeric or terminal region. So it is called as a random synapsis. So if the synapsis occurs starts at any part, then it is called as a random synapsis. So in electron micrograph, the synaptonymal complex appears as a linear complex of three parallel proteinaceous strands. So three parallel proteinaceous strands are going to observe, and these are situated between the pairing of a homologous chromosome. So these are the homologous chromosomes, and in between we are going to observe three parallel proteinaceous strands, and there is a dense layer at the center called as a central element. So these are the central elements, and there are two dense layers on either side of a central element and called as a lateral elements. So we are going to observe the central elements and also lateral elements. So the central and lateral elements are held together by a means of fibers or elements called as a transverse fibers. So these are the transverse fibers or a transverse filaments. So the synaptonymal complex helps in the establishment of a platform for a future genetic recombination. So this synaptonymal complex helps in the genetic recombination, and synaptonymal complex helps in a gene-to-gene pairing. It helps in keeping the paternal and maternal chromosomes together during a pairing. So next is a packaging. So during this stage the pairing is completed. So in the packaging, pairing is completed. In the early packaging, the chromosome pair have a longer chromosome which are thread-like. The paired chromosomes during packaging are called as a bivalents. So in the packaging stage, the paired chromosomes are called as a bivalents, and the chromatids of a bivalents divide longitudinally, and thus each one contains two chromatids, and this stage is characterized by the crossing over. So in the diagram you are going to observe the crossing over, and it is defined as an exchange of a genetic material between non-sister chromatids of a homologous pair of chromosomes. So crossing over is the exchange of the genetic material between the non-sister chromatids of homologous pair of chromosomes. So during a crossing over, first breakage of a chromatid takes place and is followed by reunion of separated chromatids between non-sister chromosomes. And crossing over is the most important phenomenon where it helps in genetic recombination and it leads to variation, which are tools for the evaluation. And the nuclear membrane is present, and also here the nucleolus is enlarged. So the nucleolus is enlarged. So next is a diplotin. So the diplotin chromosomes are condensed and thick. So compared to the packaging, in the diplotin we are going to observe the very condensed thick chromosomes. The crossing over is completed. The bivalent or homologous chromosomes start separating from each other. So here the bivalents are going to separate from each other, but at the point of a crossing over, they held together by a constriction called as a chiasma, and the chiasma is defined as cytological expression of a genetic crossing over. So where the crossing over occurs or the region of a crossing over is called as a chiasma. In the beginning, chiasma are more and reduced during a end due to the process of fertilization. And the nucleolus and also nuclear membrane are also present in a diplotin. And the last one is a diakinesis. The bivalent chromosomes at diakinesis exhibit plus multiplication or Y or ring shape, and terminalization, that is terminalization, is completed at this stage. At the end of a diakinesis, the nuclear membrane starts disintegration. So this is the end of a diakinesis. We never see the nuclear membrane. The nucleolus also disappears. The homologous chromosomes, that is maternal and paternal, are a short and thick, which have exchanged the genetic materials. Finally, the chromosomes are set free in the cytoplasm. So this is regarding the substages of a prophase first. So next is a metaphase first. So the metaphase first is characterized by the equatorial arrangement of a chromosome. So we are going to observe the equatorial arrangement of chromosomes. The chromosomes are arranged on the equator of a cell and forms a plate-like structure called as a equatorial plane. So the chromosomes, they are going to form a plate-like structure called as the equatorial plane. And the arrangement of chromosomes on the equatorial plane in a metaphase first is different from that of a metaphase of the mitosis. In this stage, the centromeres are towards the poles and chromosome arms are towards the equator. So the centromeres are towards the pole and the arms of the chromosomes are towards the equator. But a reverse situation is seen in a mitotic metaphase. The chromatic apparatus become well developed in animal cells. Asters, spindle fibers are also formed in animal cells. But in case of a plant cells, the spindle fibers are the only structures which form achromatic apparatus, and the spindle fibers are get contact with the centromeres. So here are the spindle fibers and they going to contact with the centromeres. The characteristic feature of a metaphasic chromosomes at the end, which is nothing but division of the centromere, which do not going to occur. So here the centromere do not divide. So next is anaphase first. During this stage, the movement of chromosomes takes place towards the opposite poles. During anaphase first, the homologous paternal chromosome move towards one pole and the maternal chromosome move towards the opposite pole. So here the paternal and the maternal chromosome move towards the opposite poles. Due to the absence of a centromeric division during the anaphase first, the entire chromosome move towards the opposite pole. So there is no division of a centromere occurs in case of a meiosis. So the entire chromosomes are going to move towards the opposite poles and they are called as a sister chromatids. So thus there is reduction in a number of chromosomes in a daughter cells which become haploid. And the three types of spindle fibers are extended from one pole to another. So next last one is the telophase first. So the last stage of meiosis first is the telophase first, and during this stage the bivalent chromosome become arranged at the opposite poles. So the chromosomes are going to arrange at the opposite poles and the chromosomes show uncoiling and uncondensation and form a network like chromatin. So here we are going to observe the chromatin networks. And the nuclear membrane reappears, nucleolus also reappears. The achromatic apparatus, they undergo dissolution and the above events are followed by the division of the cytoplasm called as a cytokinesis. So here successive cytokinesis and also simultaneous cytokinesis, these are the two types of cytokinesis. So when the division of a cells into two takes place immediately after the telophase, the cytokinesis is called as a successive type. So when the cell is going to divide into two immediately after the telophase, then it is called as a successive type of cytokinesis. So when the division of a cell is not found after the first telophase and it takes place at a time after the meiosis second, then it is called as a simultaneous type of a cytokinesis. So these are the two different type of a cytokinesis. Further, cytokinesis may be centripetal or centrifugal. After telophase first, the cell undergo resting stage called as a interphase. So again the interphase stage is going to follow. So this is regarding the meiosis first. So meiosis first, and by this we came to know about the meiosis first, that is it involves prophase first. In the prophase first there are 5 substages we have discussed, and also metaphase first, anaphase first and telophase first. So in the next class let us know about meiosis second. Thank you.